Okay, let's continue with our discussion of details about the sickle cell anemia. So we have previously seen that when a protein is formed, then its biochemical and structural properties or behavior within the cell actually plays a very big role in producing the physical characteristics of the phenotype of the individual. Now, when a mutation occurs and that mutation alters the gene, what will happen is that mutation may modify or even eliminate the encoded protein which is supposed to be encoded by that specific gene, and that protein's elimination will usually result in elimination of the function of that protein and therefore subsequently cause an altered physical characteristic or the phenotype of the organism, in this case the individual. Now, sickle cell anemia, how does it result? It results from a mutant form of hemoglobin, and this hemoglobin, we say that is the protein which is involved in transportation of oxygen from the lungs into the cells in our body. Now, hemoglobin as a protein, it is actually made up of two different polypeptides, namely the alpha globin and then the beta globin. And this alpha globin and beta globin are each encoded by two different genes. Now, each hemoglobin molecule which is functional will usually contain two alpha globin and two beta globin molecules. So, when there is a change in the hemoglobin, what happens is that the mutation that results in the change in hemoglobin is traceable or can be attributed to a mutation in the gene coding for the beta globin part of the hemoglobin. And this change or this mutation in the gene coding for the beta globin part of hemoglobin, it causes an amino acid substitution in one of the 146 amino acids within that protein. Considering that hemoglobin will have a number of amino acids, quite a number of them. Now, when there is a change in one of the amino acids within hemoglobin, that would result in changes in the structure of hemoglobin, and therefore it will result in that sickle cell anemia that results from that mutation. Now, the exact point at which the change in hemoglobin occurs when somebody has sickle cell anemia is in the nucleotide coding for amino acid number 6 in the beta globin of hemoglobin, and that nucleotide that codes the exact amino acid which is supposed to be at number 6 is supposed to be glutamic acid. But in the case of the mutated form of hemoglobin, that glutamic acid is changed into another amino acid which is valine. Now, remember, we have said that in this case of sickle cell anemia, there is only a change in one of the 146 amino acids, meaning that the remaining 145 amino acids within hemoglobin are actually not altered, they remain intact. Now, when an individual has two mutant copies of the gene coding for the beta globin of hemoglobin, then such an individual will suffer from that condition which we are calling as sickle cell anemia. Now, this beta globin, which has had a substitution of glutamic acid to valine at amino acid number 6, what happens is that the hemoglobin molecules in those red blood cells of those individuals, they tend to polymerize or to stick to each other. And this polymerization of the hemoglobin molecules within the individuals having sickle cell anemia will usually occur especially when the oxygen levels within the individuals is low. And these low oxygen levels within the individual and subsequent polymerization of the hemoglobin molecules will result in clogging of the blood vessels of the individual with that polymerized hemoglobin, and therefore transport of oxygen will so much be interfered with due to that clogging of the blood vessels of the individuals. Now, this polymerized hemoglobin molecule on its own, it has a tendency to form long chains, of which those long chains are responsible for the distortion of the shape of the red blood cells. Now, the shape of the red blood cells is supposed to be circular or spherical in nature, but when there is that polymerization of hemoglobin, then those long chains which are polymerized from the hemoglobin will tend to alter the shape of the red blood cells of the individuals, and therefore that distorts the shape of the red blood cell, so that the person's red blood cell have this kind of shape which we have here as the sickle cell red shape, which is present within the circulation of the individuals. Now, these altered or distorted red blood cells of the individual suffering from sickle cell anemia also have another tendency of being very fragile, and having that tendency of, or having that property of being very fragile, it means that they can easily break. And this easy breaking of the deformed red blood cells is what actually causes them to have the anemia, because they will be lacking adequate amount of red blood cells within them in order to transport the oxygen as required within their tissues. Now, this anemia, it can be possible that if there was no mutation in the gene coding for the hemoglobin, will this anemia result? It could not result, because of the fact that it is due to that mutation that then distorts the hemoglobin that will then have the anemia that results from the easy breaking of the red blood cells of such individuals. Now, the red blood cells, when they are also sickle-shaped, they have that property or that tendency to also block the blood capillaries, and in this case, that blockage of the blood capillaries actually has the tendency of causing severe pain and even damage to other tissues and organs within the cell, especially the heart, the brain, the muscles, and even the kidneys. So in this diagrammatic illustration, we shall see how the sickle cell anemia results, with a detailed look at how the gene is distorted, and therefore how the amino acids at position 6 is also distorted. Now, under normal circumstances, the DNA around the beta globin part of hemoglobin should have this sequence around amino acid number 6. So under ordinary circumstances, we have CTC here, then GAG in the case of the messenger RNA that has been formed from that DNA, and then the amino acid sequence at this position will be glutamic acid. On the other hand, with the case of hemoglobin that has been distorted in shape due to that mutation, instead of CTC in the normal DNA, we'll have a GAG resulting there. Sorry, CAC, not GAG, but CAC resulting at that position. And therefore, that CAC, the corresponding codon or the corresponding sequence in the messenger RNA at that position will be GUG. And this GUG substitution then causes the particular codon here in the messenger RNA to code for valine instead of what? Glutamic acid. And that exactly is what causes the hemoglobin to be distorted and therefore resulting into sickle cell anemia. Now, the symptoms of this disorder, called as sickle cell anemia, actually result from that change in the normal nucleotide sequence within this gene that we have been seeing here. The normal sequence, which is supposed to be CTC, but has been changed to what? To CAC as a result of a mutation. And that requires that then dictates that the resulting protein will have a distorted amino acid sequence. And this actually demonstrates the importance of the relationship that must be maintained between the genome of the individual, or the genetic structure or the genetic component of the individual, and the phenotype or the physical characteristics of the individual, especially at the protein level, so that the normal processes that occur within the individual are not distorted. And we have seen that if they are distorted, then of course it will result into disease development. So in that case, we'll have seen how the structure of hemoglobin is related to the gene sequence that codes for that hemoglobin molecule. So in the next discussion, we are going to see a few other examples of other genetic diseases that result from what? From mutations in specific genes, and the characteristic features of what those diseases are present with, and how the gene products are also altered. So we shall see that in the subsequent video. Let's stop there for now.